Hi, my name is Zhoujie and I'm from Monash University. Today I'm going to describe our JM paper entitled Ki67 deficiency impedes chromatin accessibility and BCR gene rearrangement. Ki67 is widely used as a marker for identifying proliferating cells. It was found that it functions as a surfactant to maintain chromosome structures during mitosis and is also required for maintaining heterochromatin structure during interphase. But it is largely unknown what the physiological roles for Ki67 are in vivo. To address this, we generated a germline Ki67 knockout mouse line and found that their growth development was largely normal. But when we examined the immune cell composition in the spleen of Ki67 knockout mice, we found a specific and significant impairment in the accumulation of B and T cells. The reduction in lymphocytes was not due to impaired proliferation potential of mature Ki67 knockout cells, as demonstrated here by the normal in vitro division of Ki67 knockout B cells. Instead, we discovered significant impairment at the large pre-B, small pre-B, and immature B cell stages in the early lymphopoiesis. Using a differentiation index, we revealed that impairment first occurred at the transition from pre-Pro-B to Pro-B stage. Similar defects was observed in some cell development, also not shown here. The partial blockage in early lymphocyte development in the absence of Ki67 coincide with stages where antigen-receptor gene rearrangement occur. As the proliferation of Ki67 deficient cells is normal, we hypothesized that Ki67 was required for gene rearrangement process. Using ATAC sequencing, we found substantial differences in chromatin accessibility in pro-B and small pre-B cells in the absence of Ki67, whereas minimal changes were observed in Ki67 knockout pre-Pro-B cells. A further dissecting of the specific regions that affected by Ki67 deficiency revealed a significant decrease in the accessibility at the putative enhancer region of the reg locus. This was in line with a decreased reg1 mRNA expression in Ki67 knockout pro-B and small pre-B cells, whereas the reg2 expression was unaltered. Indeed, we found gene rearrangement was less efficient in the absence of Ki67, as shown here by the overrepresentation of the germline sequence found in the pro-B and large pre-B cells, suggesting abnormalities in receptor gene rearrangement in the absence of Ki67. Lastly, we showed that the presence of pre-rearranged Ig-heavy and Ig-kappa alleles fully reversed the impairment of immature B cells in the bone marrow of Ki67 deficient mice. Taken together, our study identifies a distinct role for Ki67 in sustaining proper antigen-receptor gene rearrangement during lymphocyte development.